Okay, well, welcome to the 2021 Land Lab Lecture Series. I'm leading an inaugural studio this spring as framing a UVA satellite site, Milton Airfield, as a mesocosm for ways of knowing through situated making, where students will research, design, and build a mesocosm investigation to then challenge, interrogate, and potentially reorient through a series of exercises exploring alternative ways of knowing and creating knowledge. And in tandem to the studio, this lecture series is a parallel effort in positioning the studio's efforts on site within a history of precedence in theory and practice. And tonight we're joined by Elizabeth Hénaff, who is a computational biologist with an art practice. And her work focuses on the invisible living component of engineered landscapes, specifically the environmental microbiome. She has a bachelor's in computer science, a master's in plant biology, and a doctorate in bioinformatics. So she's quite versatile. And at the center of her research is a fascination with the way living beings interact with their environment. And this inquiry has produced a body of work that ranges from scientific articles and peer-reviewed journals to projects with landscape architects. I worked on a project with her in Guanas Canal, to artistic practice. She has made contributions to understanding how plants respond to the force of gravity, how genome structure changes in response to stress, and most recently has turned her attention to the ubiquitous and invisible microbial component of our environment. So please join me in welcoming Elizabeth Hénaff.
Thank you so much for the introduction, Matthew, and also for the invitation. I think it's always fun to give guest lectures because it's like the best part of teaching, because you get to like share fun ideas and like have good conversation without the weight of like having to give people grades and evaluations and stuff. So thanks for this opportunity. And yeah, thank you everybody for showing up. And I feel like time almost feels really, or like online time feels, cheap because it's abundant these days. And so I think that actually showing up and participating online is something to be grateful for. So thank you all for showing up. I prepared a few ideas and projects that I wanted to share with y'all today, but I'm hoping that this can be as interactive as possible given the format. And so if you have any questions along the way, feel free to speak up and unmute yourself and just ask away. Or if it feels more comfortable to put it in the chat for us to discuss later, please do so. I definitely welcome it. So I'm going to share my screen. Let's see. There we go. Can you see my screen? Cool. I'm going to arrange your faces in a way that is least disconcerting to me right now. And do this. All right. I think we're all set up. Good. Can you see a black screen with white text on it?
Yep, all good.
Cool, awesome. So yeah, today I wanted to talk about some apparatuses and living interfaces. And so, so as Matthew mentioned, I've been trained in the sciences, both in the biological sciences and computer sciences, and in the sciences, we're really taught to remove the human being from the equation, from the process of knowing. And that manifests in a bunch of different ways. So it manifests in the way, for example, that we write in the third person when we write up a scientific article. So you'll say, the experiment was performed according to protocol XYZ instead of I performed the experiment. And so for perfectly good reasons, because, we try to run experiments in a way that they're reproducible so that they're not dependent on a particular individual to be able to get that result. But it's also really eliminating kind of a key aspect of the practice of science, is that it is individual practitioners who are pursuing this way of knowing. And those are individual practitioners who come with their own ideas, preconceived ideas, biases, intentions. And even though that's something that we try to eliminate from the process, I think that is something important to acknowledge. And that also manifests in the relationship that we have to the instruments that we use. So, the instruments that we use are considered to be kind of these impartial extensions of our sensory system, just kind of these like neutral agents that just allow us to see a little better or hear a little better or feel things in different frequencies. But those extensions of our sensory system are not impartial, just like our sensory system is also not impartial. It has very limited scope, or very well-defined scope, shall I say. And so throughout kind of my practice as a scientist, I started really realizing that a lot of the relationships that I have are more so relationships to instruments rather than relationships to the organisms that I study. And I've studied, plants and humans and microorganisms, and really kind of, even in the process of being really intentional about getting to know an organism, most of the interactions that you have are with instruments. And so kind of what I wanted to talk about today was give a few examples of different instruments that I've used, and kind of how I see them defining the relationships I've had with organisms of study, and also talking about the noise in those systems. So, when Matthew shared the brief with me for the assignment that you have for this week is, both diagramming an instrument and also talking about the noise in that particular system. And so what I wanted to do today was kind of give just kind of unpack those thoughts with relationship to some instruments that I've used. So with that, this very blurry and pixelated image is an image acquired with a supercooled CCD camera of bioluminescent plants. So these are plants that I genetically engineered to express a luminescent reporter. So they express a gene that catalyzes a reaction that produces light. This is actually the same identical reaction that fireflies use to blink. And so these plants are engineered to express these light-creating genes under certain types of conditions. And so this is called using a reporter system. So when you want to understand something about kind of the cellular function of an organism that you can't measure directly, what you can do is associate that particular function, so the expression of a gene, for example, with something that you can measure. And light is something that we're actually really good at measuring. And so in this particular case, these plants were engineered to express this luciferin luciferase gene during a particular process of their reaction to the force of gravity. So, plant roots grow down, and they just somehow know where down is, and plant shoots grow up, and they also know where up is. And so that particular process is called Graphotropism. So where growth responds to the vector of gravity. And so this was a research project that I was working on when I was a master's student, actually, trying to figure out how plants perceive the vector of gravity and also respond to the vector of gravity. And so I realized when I was doing these experiments, these experiments, like the microscope was like buried in this tiny little room in the depths of the protein analysis facility. And there was full of all these like glitchy instruments with all these like tubes coming out of them and stuff that would like hiss and purr and kind of like squeak all the time. And so I tended to do my experiments at night, and I realized that my experiments actually worked better at night. I tended to have less noise in the images when I did the experiments at night, and I couldn't quite figure it out. And I was like, well, maybe it's just because I'm a night owl and I actually really like the time that I think best is when I've had 3 cups of coffee and it's midnight and I'm all by myself, and that's just why like my experiments are working better. But actually, I was talking to the manufacturer of this particular microscope, and they were like, "Oh yeah, it makes sense because the cosmic particles, you have less cosmic particles at night because this camera lens is so sensitive that it registers alpha and beta particles that are being emitted by the sun, and that will traverse this like tiny little square, which is the sensor of the camera, and register as white dots, which I was perceiving as noise, as like a glitch in my instrument. And so my experiments worked better at night when I had an entire planet between me and the sun. And the planet, Earth, would absorb some of those particles, and my experiments weren't as glitchy. And so I think this is kind of the first instance in which I realized that this instrument, beyond doing what it was supposed to do, which was take pictures of my little genetically engineered plants, it was also giving me this idea of situatedness on a cosmic scale. So it gave me a sense of my place on this planet and the relationship between this planet and other planets like the sun, and this kind of malfunction of this instrument that was supposed to be measuring something infinitesimally small in a very like localized spectrum, was also registering something that was infinitesimally large on a cosmic planetary scale, and that was supposedly an error in the function of this instrument. But for me, gave me a lot of additional information. This information about my place, my place in the cosmos, if you will. And so I think this was kind of the first instance that I really thought about the relationship, between myself as a scientist and the instrument, and how actually it is a lot maybe more subtle and complex than the relationship that the instrument was designed for. And so Karen Barad has written a lot about the role of the instrument in the sciences, and I think I sent you all a link to an interview of hers as potential reading for this context. But she's she's trained as a physicist, but she's also a feminist theorist and a scholar, and she's written a lot about the theory of science. And so, in particular, I really like this passage, and so I thought we could read this together before we start thinking about more instruments. So she says, apparatuses are not inscription devices, scientific instruments set in place before the action happens, or machines that mediate the dialectic of resistance and accommodation. They are neither neutral probes of the natural world, nor structures that deterministically impose some particular outcome. So in my case, my instrument, my microscope that was designed to measure the infinitesimally small was actually also measuring, the cosmically large. So then she continues to say, importantly, apparatuses are themselves phenomena. For example, as scientists are well aware, apparatuses are not preformed, interchangeable objects that sit atop a shelf waiting to serve a particular purpose. They're constituted through particular practices that are perpetually open to rearrangements, rearticulations, and other reworkings. And so then she talks a little bit about how there's all of these, what she has defined as intra-actions between the apparatus and the practitioner, how the practitioner influences the apparatus and the apparatus influences the practitioner. Also these intra-actions between apparatuses, between apparatuses, and how these different instruments can also influence each other. And so in her perspective, there's this kind of porosity in the boundaries between the practitioner, the apparatus, and the other apparatuses that it interacts with. So she concludes this paragraph saying, boundaries do not sit still. So I encourage the reading of this article if y'all are interested in kind of thinking through the theoretical frameworks around this. So one of the apparatuses that I'm most familiar with is the apparatus or process of DNA sequencing. So this is a reasonably recent technology. It was DNA was discovered in the 50s. And we can consider DNA sequencing as somewhat of an instrument, even though it's a process. So you start with a biological sample, and then you manipulate that biological sample with a whole series of steps that are aimed to break open the cells of that sample, separate out the genetic material, so the DNA, from the rest of the cellular debris, proteins, and other cellular contents, and then extract that DNA into solution. And so on the left here in this image, you can see all the tubes that correspond to all of the different steps that led from the extraction of this biological sample here, taken with a swab, to the purified DNA here at the bottom. And then we can sequence that DNA, which generates a digital readout of the physical molecules that we extracted from that biological sample. And so as such, this is kind of an instrument similar to the microscope that allows you to get a reading of the sequence of nucleotides, or these letters, A, C, G, T, in a digital format that corresponds to the molecular sequence encoded by that particular organism. This feels like a really powerful instrument because DNA is the common information storage molecule used by all living organisms that have been studied or characterized to date. And so in a certain way, it's a way to read out the program, if you will, that has led to the formation of that particular organism. So DNA was discovered in the 50s by two scientists, Rosalind Franklin, and then further, those discoveries were furthered by Barbara McClintock. So Rosalind Franklin was a technician working in the Watson and Crick's lab. Watson and Crick were credited for the discovery of DNA, but she actually did all of the experiments for it. And so this image on the right here is the X-ray crystallography image that led to whose shape led to her conclusion of the structure of the DNA molecule. So as you'll see, this seems like a very cryptic image to most of us who are not trained in reading X-ray crystallography. But to a practitioner of this technique, this is not at all noise. This is very much signal that leads to a very specific conclusion. And so DNA as the kind of information or identity coding molecule has really made its way into our kind of common understanding of the nature of identity. And there's a strong kind of cultural trope around this idea of DNA being equated to the notion of identity. And so you can find all sorts of examples of using the expression "it's in my DNA" to mean that it's something within my nature, that it's my identity. And so I think when we're thinking of veracity of instruments, DNA is considered to be something that is beyond question, in a certain way, like uncontestable. That it kind of encodes, some kind of deterministic, deterministic form of identity. But what I wanted to talk about today is this instrument of DNA sequencing, and actually the good amount of noise that is in that particular practice, and kind of push back against this idea of it being a completely true readout and determinism of identity. So when we use DNA sequencing as a way of knowing, what's most interesting really is when we're able to compare DNA sequences between each other. So compare one organism to another. So what that looks like is taking these very large flat text files and trying to find differences between them. In this particular case, the image on the left and on the right, one of them is a tumor genome, and on the right is the genome of the same patient, of a healthy tissue of the same patient. And so a lot of the exercise in personalized medicine is, in particular in the context of cancer research, is the oncologist will prescribe a whole genome sequencing of, say, an excised tumor and a healthy tissue of the same individual. And the question will be to look at the kinds of differences that you can find. And those differences might be causative of the cancer. So maybe some of the original mutations that led to a whole cascade of genetic changes that led to the cancerous state. Or those mutations might be a consequence of the cancerous state. But in any case, they might inform decisions by the oncologist for a particular course of treatment. So it's kind of like looking for a needle in a haystack to be able to find the significant differences between these two text files. And so the human genome is 3 billion bases long. So that's 3 gigabytes. So imagine a 3 gigabyte text file of sequencing data, and then you want to find the places that are similar and that are different. And so the kind of differences that we might be looking for can be point mutations. So looking at finding sequences that otherwise are identical, but that are just different in one particular location. Or they might be looking for much bigger mutations. So an entire chunk that has been rearranged, inserted in a new place, or removed from that place. So those would be translocations, insertions, or deletions. And so using this example, with English text here, we can see how these point mutations or these large translocations might completely change the meaning of the interpretation of this particular text. And so when we're thinking about the design of an instrument, so here in this case, maybe the instrument is not only the DNA sequencing mechanism itself, but also the algorithms used to analyze it. We could diagram out, the way of looking for these differences, but a really important part of this approach is also filtering out the noise. So you could imagine that a difference in just one letter might be an error in the sequencing process. So how do you determine that this difference in this one particular letter is actually a real signal versus a noise? Same thing for this. So this might be, a computational error that when you're reconstructing these sequences, that you introduced a repeated piece in a place where it should have been elsewhere. So when we're thinking about signal versus noise ratio, a lot of the time when we're running these analyses, the signal is actually a very small fraction of the differences that we're detecting versus the amount of noise. And this matters greatly depending on the scale at which you're evaluating these differences. So say in the case of this patient, we're looking at a time scale of 1 lifetime. So differences that have arisen between two cellular or 2 organs, let's say, within a same patient. And so differences that we see, even slight differences, might have a really strong impact because you really expect for those 2 cells' genomes to be absolutely identical. But if you're observing differences across 2 generations, so maybe between a parent and a child, those differences might carry less weight or be less important because you already expect for there to be differences from one generation to the next. Or for example, if you're comparing two plant varieties that you know have diverged, 100 or 10,000 years ago, the differences that you observe, the kind of threshold between signal and noise, and those differences that you observe is going to be a lot more strict, because you do expect there have been many different mutations across the different lineages of these two plants who shared a common ancestor maybe thousands of years ago, but since then have diverged. And so when we're talking of signal to noise ratio within our instruments, I think it's important to consider what scales you're making these evaluations at. Given that the time scale, or the scales in time, or maybe space, or even size and resolution actually really impacts what you call signal and what you call noise. Also, when we're talking about genetic information, it's very much entangled with socio-political constructs within our societies also. So the word mutation carries a very negative connotation because we assume that that's like a difference from the norm. But what we have defined as the norm is very much a social construct, just as well, or maybe even more so than a biological one. So all of human genomics studies are based on the comparison between individual sequencing data and the human reference. So there is one single human genome that is considered to be representative of the human genome. This was a genome that was published in 2001, and there was the amalgamation of 5 different individuals' DNA. And so those individuals were, I believe, 3 men, 2 women, 1 African American, 1 Hispanic, 1 Asian American, and 2 Caucasians. They were chosen as representatives of the human genome. And so even though there was an attempt made at diversity, that is definitely not representative of the breadth of diversity of the human genome. And so I think that it's interesting also, or it's important also when we're thinking of design of our instruments and calibration of our instruments, to be very cognizant of the fact that there are a lot of social and cultural entanglements in our definition of these calibrations. So what do we call normal? What do we call mutant? What do we call signal? What do we call noise? What do we call divergent? Or what do we call, how do we calibrate these instruments? I'm going to shift gears and talk about different organisms now. Are there any questions or comments or reactions at this point? No? Cool.
Continue on.
What's that?
Just saying, continue on.
Cool. So, this instrument of DNA sequencing has been really revolutionary in our understanding of ourselves as human beings. And so that is what has basically, brought on the advent of the whole field of precision medicine, so genomic-based medicine. But we can also use this instrument of DNA sequencing to study non-human organisms. And particularly, it's a really interesting tool to study microorganisms. So microorganisms are kind of tricky because they're invisible. They're really small. We can't see them. We can, see them. Some of them in a microscope if you have a sufficiently powerful microscope. But they're actually, really, really elusive because they're hard to observe directly. And so a lot of what we know about microorganisms is through culturing microorganisms. So taking a sample, streaking it out in a petri dish, putting that petri dish in an incubator at a particular temperature, and waiting to see what's going to grow. And there's some organisms that do well in growing in a lab. In particular, human-related pathogens or human-related microbes are generally pretty easy to culture in a lab because they like kind of, constant temperature, like fixed environments. But environmental microbes, so, microbes that we find in soils or in other types of environments, tend to be really hard to grow in the lab. And we call these recalcitrant microorganisms, which I think is just makes me think of this like grumpy little microbe who like refuses to grow in a petri dish. And I really love that expression. But so these recalcitrant environmental microbes are really hard to get to know if the only way that you can get to know a microbe is to grow them in the lab and see what they do in a petri dish. And so that's why the advent of DNA sequencing as an instrument and as a way of knowing has been really revolutionary and exciting, because you can take a sample from an environment, so say from like a forest soil, like the forest soil in this image, and extract DNA from it without having to go through a culture step, sequence that DNA, and then you can ask a bunch of questions of that data, like what kind of microbes are there, what kind of species, what do they do, what kind of metabolism do they encode, and so you completely bypass that step of culture. And so that allows you to get to know a lot of environmental microbes that you wouldn't have been able to know otherwise. And so that has kind of brought on this entire explosion of the field called metagenomics. So genomics being the study of the genome of one organism, and then metagenomics being the study of the genomes of a whole population of organisms, like the whole population of microbes. And so what this has taught us is that microorganisms have really influential relationships with the macroscopic multicellular organisms that they interact with. So for example, there's a paper that showed that you can alter the flowering time of plants by changing the microbial composition of the soil in which they're growing. And so this was kind of a big deal in the plant biology world, because flowering time was thought to be the like canonically genetically determined phenotype. So, plants have different strategies for reproduction. They flower at different times of the year. And that was thought to be just like strictly determined by their genetics. But what we found out was that that was determined not only by the plant's genetics, but also by the genetics of the microorganisms which they were interacting with. And so this is the case for plants, but this is also the case for mammals, including humans. So we're pretty familiar with the idea of like the gut microbiome being really important for health and well-being, and disruptions in the gut microbiome function have all sorts of associations with different types of pathologies, like IBS and Crohn's disease. But we're also finding that the interactions, or our interactions as humans with microorganisms, can affect a lot more subtle phenotypes, like mental health and well-being. So, the gut microbiome, turns out, can influence the production of serotonin, which is a neurotransmitter used in the brain, so a molecule used as a communication mechanism between neurons, and deficiencies of which are associated with conditions like anxiety and depression, schizophrenia, and things like that. So these are just a couple of examples, but there's many, many examples that show that the phenotypes or physical characteristics of macroscopic organisms like plants and humans is due not only to the genotype or genes that given organism encodes, but also the genotype of the microorganisms with they interact. And so if we go back to this idea of genetic of genotype, or genes being encoding the notion of identity, then that kind of begs like a redefinition of the notion of the individual, in particular kind of the definition or boundaries of or genetic boundaries of the individual, and to broaden that to include not only the genetic identity or genetic makeup of the macroscopic individual itself, but also of the microorganisms with which it lives in symbiosis. And so there's a term for that, which is the holobiont. So the term holobiont encompasses the notion of both host and symbiont to kind of redefine the notion of the individual. And so just as genomics is the study of the set of genes of a particular individual, we can define the Holo Genome. So that is the set of genes encoded by a particular holobiont. And so this is the case where an instrument like DNA sequencing has really given us an insight that kind of defines a field, but also redefines these kind of socio-cultural constructs that are really deep and embedded, such as the construct of the notion of identity. And so I think we can kind of carry that with us and then think about, all of our interactions. How could they be redefined if we thought of ourselves as holobionts instead of just macroscopic organisms? And so a lot of the work that I've been doing at my lab is trying to understand the relationship between humans, the human microbiome, the environmental microbiome, so the sets of microorganisms that we interact with on a daily basis, and then how design decisions for the built environment influence the environmental microbiome. Because ultimately, if the design decisions of architects, city planners are kind of sculpting the environmental microbiome, and the environmental microbiome strongly influences the human microbiome, then the microbiome is kind of this continuum that allows us to link individual and environment and the relationship between those two. And so to start teasing that out, on one hand, we need to make maps. So just to understand what microorganisms are present in their environment. And so this is a project that we worked on a handful of years ago now, characterizing the microorganisms of the New York City subway. And so if any of y'all were in New York in 2015, you can go in and view this interactive visualization and see what kind of microorganisms you're interacting with as you're taking the subway. The main takeaway of this study is that most of the microorganisms that are in the subway are brought there by human activity. So they're either human-related microorganisms or related to the fermented foods that we eat. But another really interesting finding is that we found a higher proportion of multi-drug resistant bacteria in subway stations next to hospitals. So we know that multi-drug resistance is a big problem in hospitals. So these are human pathogens that are acquiring resistance to multiple different antibiotics, and they become very difficult to treat. And so the acquisition of these types of pathogens is a really big problem in hospitals. So you might go to the hospital for one thing and then stay in the hospital for one of these particular multi-drug resistant infections. And so understanding that there is kind of a porosity in the boundary between hospital and city, that can directly inform design decisions and try to think, rethink the entrances and exits to hospitals. So maybe we need to have copper flooring, which is a strong antimicrobial agent, or apply negative pressure to hospitals so that when you open a door, the air always goes in instead of going out. And so one of the main tools that we used for this study is the swab that we are all now incredibly uncomfortably familiar with, because we've had swabs stuck up our nose way too many times for COVID tests. And so in thinking about this instrument as a tool and as a way of knowing, I think it's interesting to think about the relationship and scale between the tool that you're using and the thing that you're studying. So in this case, we were using a swab, so that's like at the resolution of a centimeter, to studying, all of New York City, which is at the resolution of kilometers. And so in thinking about relationship to instruments and what kind of noise can come out of the systems, just as we talked about timescales earlier, I think it's important also to think about scales in space, because if the sampling instrument that you're using is so small, then you have to make decisions as to what information you're going to collect. So am I going to take a swab of the sidewalk, of the side of a building, of the inside of the subway, of the hand railings in the subway? Like, what is going to be a representative sample of my topic of study, which is the city in this case? And so I think thinking about the relationship of scale between your instrument and your topic of study is also really important. And so we had another project that arose kind of this acknowledgement of the discrepancy in scale between our tool and our subject of study. And so for this project, the subway project, we realized that there were 462 subway stations. And so we take 3 samples each for biological replicates, and then we take 3 samples, repeat that 3 times for technical replicates, and we did that for 3 years. So we ended up with 12,000 odd swabs, which cost us about $2.5 million in sequencing costs. And so that kind of made us realize that we really needed a different kind of instrument to study this particular, to conduct studies at this scale. And so we really racked our brains to see, like, okay, how can we get a more or less granular resolution of sampling of a whole city? So, like, how could we get samples from a whole block at a time, for example? And so we realized through a whole process of like very bad ideas, we happened on the idea that honeybees are actually doing pretty much what we were asking our undergrad students to do, which were leave the lab in the morning, run around the city and like gather stuff, and then come back to the lab in the evening. And so we piloted this project to see if we could use the debris at the bottom of hives, of urban beehives, as a representative sample of the metagenomic or microbial information of a particular area. And so we did a whole pilot study to like make sure that this wasn't, that we weren't getting just bee DNA or just DNA from the beekeepers' hives. And I'm glad to discuss the details of this experiment further. But ultimately, we figured out that the debris at the bottom of the hive is a really good representative sample of the microbial environment. And surprisingly so, not just the microbial environment of what you expect bees to be physically interacting with, like plants, but also the microbial environment of, say, a body of water, if there happened to be, or aquatic environments, if there happened to be a body of water nearby, human-related microbes, animal-related microbes. And so we realized that what was happening is that, bees, as they're flying around and foraging, searching for food, they're traversing what has been kind of dubbed as microbial clouds. So, a microbiome isn't just like a discrete entity. It's actually just a general continuum. So you can think of it kind of like temperature, like you can measure temperature at different locations, but the metric of temperature exists as a continuum between all of those locations where you're measuring it. And so there's research that has shown that, we all kind of exist in the world surrounded by our microbial clouds. So we each have our own microbial cloud around each of us that leaves traces in places that we go. And we can also think of the microbial cloud of a park or of a forest or of a lake or a pond. And so these bees are traversing these microbial clouds, or aerobiomes, which is a real term that is used in science that I discovered recently, and which is the microbiome of aerosols. And so these bees are traversing these microbial clouds, and collecting this microbial information, and then eventually shedding it as they enter into the hive. And so we did this study in a bunch of different cities. We piloted in Brooklyn, did a bunch of different cities. And one of the cities that we had the most density of urban hives was in Tokyo. And so we did this study where we looked at the microbiome of a bunch of different hives, and we were actually able to establish correlations between the distance according to microbial diversity and physical distance. So the heat map that you're looking at here on the right is a heat map that describes abundance of microbes. So yellow color being high abundance and green color being low abundance of this whole different list of microbes here on the y-axis, on the vertical axis, and then a bunch of different beehives here on the x-axis, on the horizontal axis. So you can see that this set of beehives here, for example, all had a high abundance of this microbe, but a low abundance of this microbe that occurred in these two hives. And so this kind of quantitative biological metric gave us, was correlated to the quantitative kind of geological, or I guess geographical metric of distance, and gave us an idea of I guess the like more high resolution microbiome of an environment. So that was, that was a much less granular sample than the individual swab, but that gave us kind of an average readout of a particular neighborhood. And so when we're thinking of noise in the system and how this instrument dictates relationships, now this was not just a relationship to a microbe, but it was a relationship to a whole other organism that was mediating between microbes and ourselves. So this was a relationship to bees, and then we were talking about the bees' relationship to microbes. And so this was, like a very proxy relationship between these different species. But, and so in a certain sense, we were getting like a bee's eye view of the city microbiome. But that really shows us that, our cities are very much these multi-species entanglements. And when we're able to kind of be able to just like open up a lens into that, I think that can be very, very rewarding, even though there's obviously a lot of noise in the system. So when considering this instrument, we need to consider the different personality of the different beehives. So, if you consider a beehive as being the organism, kind of a superorganism, different beehives have different personalities. Some of them are a lot more adventurous than others. They travel further. Some of them are more aggressive than others. You can tell, kind of how I guess ambitious they are with respect to how much comb they're building, how much honey they amass. And so in this case, the noise in our system wasn't really, kind of a mechanical glitch in our instrument, but just the different personalities of the bees that we were working with. How am I doing on time? Am I okay?
You might be running up against it pretty soon.
Okay.
Yeah.
Cool. So these types of projects of mapping microbiomes specifically focus on understanding the relationship between built environment and the built environment microbiome is the kind of projects that we work on in our lab. And so these are all mostly focused on scientific output and like scientific publication output. But I think it's also really interesting to acknowledge the fact that, the relationship that we have with these invisible organisms is very much a relationship to the tool and the instrument. And to think about, how can we, or acknowledging that and kind of owning it, how can we design interfaces or interactions with these invisible microorganisms, specifically with the explicit purpose for collaboration. So as Matthew mentioned, we worked on a project together looking at the Glanz canal microbiome. And we found that the microorganisms in this contaminated canal are able to degrade the toxic compounds that they're challenged with. And so this was a project that I made, which was called a gene zine. And so this was a zine that like kind of like an artist book that was made as a genomic note from the underground serving the subculture of multi-species collaboration. And so the way this zine was designed is that it was embedded with DNA that I extracted from the Gowanus canal microbiome. And so the idea is that you can tear out that piece of paper and embed it at a location that is in need for remediation. So microorganisms are able to absorb DNA from the environment and kind of hot swap that in and start using it. And so the Gowanus canal has had 150 years to adapt to that particular cocktail of industrial contamination. And now that knowledge that is encoded in their DNA sequences can be shared as this kind of practice of collaborative survival through this zine publication. Another project that I worked on was a collaboration with the architecture studio called The Living that is led by David Benjamin and Kevin Slavin, who was professor at the MIT Media Lab at the time. And so this was a commission for the Storefront for Art and Architecture, where they tasked us to think about what it would look like on a human scale or an architectural scale if you were to design for microorganisms. And so this was an exhibit at a gallery in downtown Manhattan that has these big kind of swiveling doors. And so we designed these bioreceptive panels to be mounted on those doors as a way of creating microenvironments, but that were visible on the human scale. And so in general, what I've been thinking about these days really is this idea about how the instrument mediates the relationship. And so on the left, we have the swab that we're all uncomfortably familiar with, and it has this really negative connotation of, disease, contagion, and that it makes sense because this object is a clinical object. It was designed for a clinical context, specifically in relation to human pathogens. But its role really is to collect the biological sample, to gather organisms. And the image that we see on the right is doing exactly the same thing. So, grabbing a handful of soil is also taking a microbial sample, but the image on the right has these, positive connotations of, earthiness, groundedness, connection. And so really the interface really matters between microorganisms and ourselves, given that that's the relationship that we have. And so, in the context of this pandemic, I've been thinking a lot about it. And, I think on one hand, this kind of emerging idea of living collaboratively with microbes has really taken, 10 steps backwards, given the extremely contentious relationship we have at the moment with the unseen biological component of our environments. And that is manifested by all of these interfaces that we've now become familiar with. So, the masks that we wear, the anxiety that we feel when somebody is standing too close at the grocery store, the new rituals that we've developed, of washing your hands when you come back home. And also, I think that it's really taken a step back with respect to the industrial scale cleaning practices that are being put in place in our public spaces, in our schools, in our workplaces. Just when I go to campus, the whole school smelled like disinfectant. And so I think we've really taken a step back in that idea of collaboration with microorganisms, but I think we're also in an interesting place because now we have this shared collective experience of having a really explicit relationship with the invisible microbial component or invisible biological component, because viruses are not microbes, of our environments. And so I guess what's been interesting to me these days is to kind of contextualize this historically and realize that we have never not interacted with microorganisms. And it feels like a really new, contentious relationship at the moment. But I find solace kind of in thinking about the fact that that has never not been the case, and that we've, co-evolved with these microbial metabolisms ever since, multicellularity has evolved. And that has manifested in a bunch of ways, and we can find artifacts that correspond to that. So, we made microbial maps of New York City, but, this was a map made of in London during the cholera outbreak, where Dr. John Snow identified all the houses that had a case, and using this data visualization was able to identify that all of these houses shared the use of a particular well, and by closing that well, was able to curtail the epidemic. Or, for example, this is the oldest bioarchaeological record of the use of fermentation for food preservation. So these were some troughs that were dug in a rock that were found in Sweden that were dated to 9,000 before the current era. And so in these troughs, they found these fish bones that had been compressed, like squashed. And so the hypothesis of these archaeologists is that the deformation of those fish bones, so you see the 9,000-year-old squashed fish bone here compared to a present day same fish bone here. And so their hypothesis is that the only way that it could be deformed in that way is in an acidic environment. And so one of the characteristics of fermentation is producing an acidic environment. And so this is an example of how kind of the materiality of the bone, but also of the rock and of the trough that was made, was what was mediating this relationship with invisible microbial components. And you find that a lot also. I mean, we've been using fermentation as a technology for a very long time, even before the understanding of microbes, like the mechanistic understanding of microbial fermentation. But traditionally, the understanding is that the property of that transformation is a property of the vessel used. So not necessarily of the substrate itself, but more so of the vessel or of the container that's used. And so if you think of all the containers that we've made for fermentation, wine jars, or the fact of burning kimchi, these are also interfaces that we have with the invisible biological world. And so I just like to leave you with one example that I find to be really compelling. So there's this hypothesis that rain dances work through the phenomenon of bioprecipitation. And so there's a particular microbe called Pseudomonas syringae that produces a protein that is able to nucleate ice crystals at warmer temperatures than normal. So the way clouds form is that water molecules in the air will nucleate around a piece of dust or a piece of ash and create crystals, then create clouds, and then that ultimately leads to precipitation. And so these microbes can create this compound that is able to do that at warmer temperatures than normal. And so this is what's used to make fake snow, like in snow cannons, like on ski slopes, when they blast out snow onto the ski slope so that you can ski out of season. And so this is the same protein that's used for that. And so it's a plant pathogen. And so the idea is that these rain dances that last for days kick up this microbe and the proteins that it's making, and then after a while float up to the particular right spot in the atmosphere to start nucleating ice crystals when it wouldn't have happened otherwise, which creates clouds, which ultimately leads to precipitation. And so in thinking about, as you're thinking about instruments this week, I also invite you to kind of broaden your concept of instruments to include, many different types of interfaces and interactions, and also kind of the ritual also being a form of interface and interaction. And so I would love for, us in general to collectively think about what are the new rituals that we can develop going forward to promote the kind of collaborative relationships that we would like with the invisible microbial or invisible biological world around us. And obviously all of these projects have been collaborations that I owe many thanks to many people for. And I will stop sharing right now.
Wonderful. Thank you, Elizabeth. Really exciting. I wish we could actually have audio applause, but that seems to be beyond students' capabilities. I think we can go ahead and open it up to questions.
Yeah.
Well, I'll go ahead and start it off. Maybe people are shy. And again, you're welcome to put it in the chat. It can be a bit easier sometimes. I mean, it goes without saying, but great presentation. I love the topics, the sort of vested interest in a lot of these things. And obviously we've worked together, so I'm a bit biased. But yeah, sort of, I mean, kind of in response to kind of some of your earlier images and in direct sort of response to kind of the assignment for the studio, I'm curious if you have like I don't know, a story as far as kind of best discovery through sort of misuse of a tool or an instrument, or sort of like creative misuse, and how that might have, I don't know, revealed something new or unanticipated.
Like personally or in the history of science?
I was saying personally, but if you want to go to history of science, that could work too.
I think personally, so there was this one, I mean, this is kind of getting in the weeds, but there was this this one project that we were working on. I was developing this algorithm to identify particular types of sequences in plant genomes. So these transcription factor binding sites, which are where the proteins attach to the DNA molecule to then transcribe a gene. And so we were looking, you know, just writing an algorithm to like recognize that particular sequence. And then, and we got the output and it looked super glitchy. It was like that sequence, but like repeated a bunch of times, like 5 times, 7 times, 15 times. And we were like, oh man, that's weird. Like there must be a glitch in this, like what's going on? And so we were about to throw the data away and just like start over. But then we were like, okay, wait, no, let's take a little closer look at it. And so then started poking around and realized that that it wasn't a glitch, that it actually was this particular sequence that had been repeated multiple times. And so ultimately, we found out that, or we figured out that the reason it was repeated multiple times was through a phenomenon of transposition. And so what the discovery was that the fact that it was repeated multiple times meant that that particular sequence, which acts kind of like an on-off switch for genes, was modular and was able to like jump around in different places of the genome. And so if you're thinking about like gene expression as kind of being a circuit of on and off switches for genes that like turn on at different times of development or like in different parts of the body, like being able to kind of modularly switch them on and off is actually pretty rad, and also from an evolutionary perspective is like a way to like induce adaptation a lot faster than if you were asking the switches to kind of arise out of mutations. That probably requires a much longer explanation. But basically the takeaway is that we were about to dump the output because I thought it was a glitch in the code that I wrote, but then looking at it a bit closer, it was actually a pretty big finding that we ended up publishing.
Cool.
And then in the history of science, I feel like is full of it, of like, you know, people leaving petri dishes over the weekend in, you know, outside on the bench when they should have been in an incubator, and then coming back on Monday, and then all of a sudden like something wild has happened. It's like that was the way people figured out how to de-differentiate mammalian cells, like Dolly the sheep. I don't know if you remember, like in the 90s or something. Yeah. That was the first mammal to be cloned from another adult mammal. And that was because somebody forgot and like left their petri dish of sheep cells out on the bench for a while, and was, you know, gonna toss them because they thought that everything was dead, but then realized that the cells looked kind of weird. And actually that like looking kind of weird was the cells having like regressed to a de-differentiated state, which enabled them to then like ultimately become a whole sheep if they're given the right conditions.
Interesting. Huh. See that, guys? Lessons to plan for failure and sort of trial by error.
Yeah, and observe things closely, because it's the observation that makes the discovery in addition to the error.
Be open for surprises and unanticipated kind of results. Looks like there's a question in the chat. What affects bee personalities?
Oh, yeah. I'm not a professional beekeeper, and so I don't feel like I have like a really exhaustive understanding of what affects bee personalities. I think it's partially habit. So bees learn to recognize their beekeeper and recognize my understanding is that more so than their appearance, it's their way of moving. So the kind of like pace and nature of their movements. So I think part of it is training, and then part of it is genetics also. And so, like sensitive beehive will often, if the beehive, divides, and you manage to collect the swarmed beehive, that daughter beehive will also be sensitive.
In the gut microbiome of each individual bee, right? Maybe.
That's personality, but that's a really good question.
It's
human personality, obviously. So.
Yeah. Any other questions?
I have a question. Thank you. That was really fascinating. I'm curious about like when you give some of the examples of work like that designers are sort of doing, and I'm curious what suggestions you have for kind of like making that jump from understanding the DNA to like actually designing something. Like personally, I feel super overwhelmed by how complicated this idea is, and I just don't even know how I would like start to design for a microbiome. So like maybe even at school where we're not able to work with professionals, like how would you even go about exploring these topics?
Yeah, I think that's a really good question, and that actually unpacks into like a bunch of other sub questions. One of them being like, what is an optimal microbiome in the first place? And I think that's a really hard thing to address, because I think just like an optimal like skin or gut microbiome is going to be different from one individual to another. I think an optimal environmental microbiome is also different. So I think it's really hard to be prescriptive, even though there are some papers that have come out that describe like really specific geni, so not species, but like the next phylogenetic level up of microorganisms that are deficiencies in which are linked to certain autoimmune conditions. So like asthma, asthma is specifically what they were looking at in the study. And so one thing that you could think about is like being like, okay, cool. So then I just need to like inoculate my environments with like species X. But I think that's actually a lot trickier. That's actually really tricky, because we know that environmental microbes, like they're grumpy, they're recalcitrant, like they're not necessarily going to just like start growing and be happy if you like spray some somewhere. And so I think that, I think it's hard to be prescriptive, but one thing that is coming out that's kind of a consensus is that diversity of microorganisms in spaces is always better for a bunch of different reasons. Because the more diverse microbiome, the more likely it is that you would have species X that is actually important for your growth and development. But also because a really diverse environmental microbiome means that it's less likely to be colonized by human pathogens. So if you think of like an analogy, like imagine you have an island where there's no bird species and you introduce a new bird species, it's gonna like have a really easy time in that environment because it's going to be able to occupy any ecological niche that it wants. But conversely, if you have an island with a bunch of bird species and you put in a new bird species, it's going to have a lot harder time because already all the ecological niches are occupied. Like the seeds are being eaten, the branches are being sat on, like all of that. So it's kind of the same with microbes. So a diverse environment is going to be less easily colonized by a human pathogen. And so really, you know, we carry potential pathogens like in and on our bodies all the time. And the pathogenicity is usually due to a factor of concentration. So it's not like the problem is that the microbe is there or not. The problem is that if it's there in like a really high abundance. So if you have a diverse environmental microbiome, then any human pathogen that, say, you bring in is going to have a harder time reaching a high enough abundance to become pathogenic. So that's the first part of the question, which is like, what should we be designing for in the first place? And then the second part of the question of like, how do you achieve that through a choice of like materials and shapes is a really open question. One of them is material choices. So there's a lot of materials that are actually designed with antimicrobial properties. So you can get like antimicrobial paints, and I feel like there's antimicrobial components in like pretty much everything that you can think of. So that is one choice that you can not make. Ultimately, I think the dream is to have like a Grasshopper plugin that would be able to predict what your microbial environment is going to be given like your 3D model and your material choices. That's really what I would love to see. But I think we're kind of far from it. But I think we're getting closer. And I think the map making exercises is really the first part, or the first place to go. So kind of understanding like, given the metrics that we have and that are currently being designed with, like metrics of lights and temperature and CO2 concentrations and airflow, like all the things that we do measure about our environments in design form, like understanding how optimization according to those metrics are then influencing this other biological metric. So I think that that's where these map making endeavors come in. And then hopefully we can figure out like how all of those relate in this like really high dimensional space. I'm working on a set of projects with the Yale School of Architecture, where we're looking at the possibility of having a green wall infrastructure being like their design being slightly tweaked to specifically inoculate indoor spaces with plant-related microbes. So there we're looking at a bunch of stuff like choice of plant substrate, but also growth substrate, and how that combination will influence the kind of microorganisms that grow in the green wall system. And then looking at the like aerodynamics of it. So like how airflow over or through growth substrate can like pick up those microorganisms. And then looking at the modeling of that airflow in the spaces, and then how organisms potentially like deposit on surfaces. So like horizontal surfaces versus vertical surfaces, different kinds of materials.
Great. Any other final questions? Is everyone zoomed out for the day?
I totally get it.
I know I am. Well, that I think we'll close it out. But thank you again so much, Elizabeth, for joining us. Really appreciate it. Wonderful. Thursday we'll have Julian Raxworthy. I guess I should put in a word for the two people that aren't in my class right now. Julian Raxworthy is going to be speaking on sort of the practices between landscape architecture and gardening, and sort of arguing that with the sort of representational turn that landscape architecture has taken, that it sort of lost touch with its sort of the maintenance side of things, and how there's quite an opportunity to kind of bring that back. But thank you again, Elizabeth, and we'll be in touch.
If any questions come up, feel free to reach out to Matthew, and he knows how to get a hold of him.
Perfect. All right. Have a good night.
Bye now.